All right. So I think the numbers as shown here are even not insufficient to tell the impact what I can access brought to the entire world. And not to mention so many speakers, I think, from this image, consisting of so many tiny, teeny icons of the global leading scientists, even the icons are tiny, teeny. I can immediately recognize many of those familiar faces. For example, Professor John Rogers, and my own good friend, Professor Li Yanxi. I'm so delighted and honored to be one of them, starting from the a new chapter of ICANX after 5 years of accomplishment, amazing accomplishment. All right, so this is my title of this presentation today, and which is about human biology. All right, and I know many people may wonder what is human biology, what you're going to talk about around today. And I ask myself the same question: what is human biology? I, like many of you guys nowadays, you go to kind of DeepSeek, ChatGPT, Gemini, or Grok to ask the question, and I did the same thing. I think they all return more or less the same information. All right, it's very diverse, very interdisciplinary, and probably starting from anatomy. That's how we begin to understand human biology, and as sort of the structure of different organs, tissues, and but also down to the genetics, molecular biology. And end of the day, that's about how we can understand ourselves, all right, human being, and why humans take at a molecular biology level. That's the ultimate goal of human biology, and that's the power. That's really the powerhouse. Eventually, that gonna drive the discovery of new therapeutics and better diagnosis, and really ushering a new era of precision health, right? If we look into the history, I think Egyptians they came up with the names of different organs and tissues of our body, and from the very early kind of Egyptian transcripts, you already recognize those words about human organs like a brain and even cerebral spinal fluid. I was truly amazed. Such a sophisticated word terminology. Even back to kind of 8,000 years ago, I think Egyptians already recognized, wow, actually, CSF traveling, flowing in our body, connecting brain to the body. So I think since then, actually, the anatomy needs to be understood by dissection. Okay, you really open a body and know what's going on inside. That has been largely prohibited due to many different reasons. However, and I think a Hippocrates, if you are a medical professional by training, you must have into your own career by kind of reading out this very famous Hippocratic oath for all medical professionals. And he's literally considered the father of medicine. And what he truly believed, all right, anatomy, which is the ancient version of human biology, needs to be and has to be based on the form of a human body. All right. So if we want to understand human biology, we have to look into our human body, human organs, tissues, and down to the molecular level. You need to look into that using the tissues, organs from our own body. Ideally, you can actually understand human biology at an individual basis. Every patient may have his or her own unique biology. That's the basis eventually to kind of individualize the precision health care, and also individualize the treatment for the patients. All right, so that's the key message, even back to thousands of years ago, and our father of medicine, Hippocrates, tried to tell us we need to look into our human body to understand our own biology. All right. So the molecular biology, modern molecular biology, it has much shorter history, but that can completely transform or usher back to that time a whole new era because you no longer just see the different structures and atomic features of different parts of our body. You really understand what happens at a molecular cellular level. And since the discovery of the DNA double helix, now we know the molecular information starting from the genetic footprint in DNA, transcribing to the transcripts, RNA transcripts, and eventually give rise to the proteins that determines how our human body actually works. But somehow there's no way, there's a huge divide between the human anatomy and the molecular mechanism if you want to understand cell by cell, molecule by molecule in situ in the human body. That has been very, very challenging. And in particular, when you're looking to the human tissue, both normal and healthy, and also the human tissues in the context of disease, for example, this is a schematic depicting the complexity of the human tumor microenvironment, a beautiful review article I read when I was the first year as a postdoc after graduating from UC Berkeley. And so first time I realized even the tumor cells are very heterogeneous, and tumor is not just a bag of tumor cells. There's so many different cell types there, and they are very heterogeneous, very functional diverse, but also they form some unique structure. They're not random in the tissue. So can you really understand the complexity of cellular heterogeneity in the context of tissue, and also connected that cellular heterogeneity kind of upper to the whole tissue organ scale in our body and down to the molecular scale that tells you how individual genes starting from the DNA to transcripts to proteins following sort of the information flow depicted by the central dogma of molecular biology, right? So if we can understand that, I really believe we're at a kind of more comprehensive, we have a more comprehensive and a much more complete understanding of human biology across all the scales, and that will give us an ultimate power to kind of provide the best individualized healthcare health management and also treatment to individual patients. Back to 2006, when I started to think about this question, and so I realized that the tools you need to understand the complexity has to be in a single cell resolution, right? But back to 2006, and literally, you cannot find a truly single cell, but also highly informative, molecular highly informative tool to understand the single cell biology. And one of my best friends, Paul Huang, who was a PhD with Dick there at Stanford, he devised this very sophisticated microfluidic system. You capture one cell and you lyse and you count one or two types of proteins. Oh, that's a science paper, but I still think this is so hard to use this type of device to understand the extraordinary complexity of the human tissue cell by cell, because you really need to look into hundreds, thousands, or even millions of single cell to understand the heterogeneous nature of our tissue and the whole entire organ, right? And also, not to mention, you need to understand the full scope of molecular biology, and one or two proteins will not tell you the full picture of the molecular biology. And back to that time, I think that's what the people can do, maybe one or two proteins, one or two genes. I was hoping to look into one specific question, the protein secreted by either immune cells or tumor cell to dictate how they talk to each other through this protein paraffin signaling. And for immune cell, turns out that tells you the actual function of those immune cells, in particular T cells. That's a language they speak, but that's also determines the actual function of those immune cells. But that even very small, very well-defined question that involves large panel of those proteins that need to be measured at a single cell level. And so I proposed this device after my first half year at Caltech learning how to perform ELISA. And seriously, it took me half a year to fully understand how to perform ELISA. But afterwards, I say, hey, ELISA or in this format, so surface based antibody microarray format, you can measure a large number of different proteins, but you would not believe you can do that in single cells. But turns out it is possible if you can miniaturize the device such that you put every cell in a nanoliter microchamber, you are able to measure the protein at almost the same concentration as in the petri dish, but the question, can you really miniaturize the DNA microarray? So I did that, not to tell you that much details, but the technology eventually, through many generations of engineering and some commercialization effort that's now commercially available at the broker. In my own lab, we'll continue to use the technologies developed in my own lab, now commercialized by broker as this isoplex isolite single cell secretion analysis system. And also over the past couple of years, we were focusing on about 10 years, we started to focus on single cell gene expression analysis and another company I co-founded called Single-Run, and using all kinds of tools to understand the single cell across the central dogma of molecular biology. And so you really learn so much biology. That's what I want to emphasize. This is just one of the examples of our own research projects utilizing single cell multi-omics sequencing to characterize human CAR T cells as engineered T cell immunotherapy to treat human leukemia. And we sequenced about like 1 million single CAR T cells from 82 patients, and which is the largest single CAR T cell multi-omics data set. And from this data, we actually learn not just the biology, but we can also correlate to clinical outcome to tell you what's the biological mechanism driving the clinical response. You learn that directly from patients, not from animal models, right? You learn that directly from the clinical trials. What I'm showing you here is the data from 2 different clinical trials. One is, so both are CAR T cell immunoclinical trials for the treatment of pediatric leukemia, B-cell acute lymphoblastic leukemia patients conducted at the University of Pennsylvania by Carl June and Steve Grubb. So we identify all different cell states upon antigen-specific activation and depicted by different clusters. But we are asking this question, okay, which state or which cluster really dictates the long-term response of those patients, since this is one of the very first CAR-T human clinical trial and started about 10 to 11 years ago. Now we can ask, what's the correlates you can find out to tell their long-term response. So counterintuitively, most T cells need to have those potent molecules produced by them to dictate their immune effector function called type 1, but we found a type 2 T cell subset that actually correlates much stronger, much more profound and with the long-term response patients. And we were asking why. So it turns out the rich molecular single cell multi-omics data can teach you directly the biological mechanism, because we can perform something like a ligand receptor interaction, say what cells are receiving those type 2 cytokine signal. And we found there are like a dysfunctional TIM3 positive cells. And once they receive that signal, they can stop from kind of further going down to the fully exhausted state, and such that the whole population can be maintained a homeostatic, the best fitness state. So that directly tells you the biology. And in the past, people always say the large-scale omics data is a good approach to generate hypotheses. But I want to just throw out this my own opinion. So if the data is rich enough and the resolution is high enough at a single cell level, you actually directly learn biology rather than still hypothesis generation, right? It's just one example to demonstrate the power of single cell multi-omics and to understand human biology. And turns out the similar discovery was made in the animal model as well. And the two papers published back to back, I think with Professor Li Tang from EPFL last year. If you're interested in this topic, you can check out both publications. However, in most cases, when you try to look into human biology, single cell analysis is not enough. In this case, this pre-infusion CAR T cells that may work, but the complex human tissue microenvironment, such as the tumor microenvironment, you do need to have the spatially resolved single cell analysis tool and at the deepest level of the molecular biology to understand what's going on. And so it turns out people indeed recognize the importance to develop the spatially resolved single cell multi-omics, single cell omics, and largely based on the single cell gene expression analysis called the single cell transcriptomics, which was chosen as the method year 2020 by Nature Methods. In that special issue, Professor Xiaowei Zhang reviewing the progress of imaging-based spatial gene expression analysis. Professor Joachim Lomberg reviewing the sequencing-based spatial gene expression or spatial transcriptomics. So we were one of the very first group realizing the importance of multi-omics spanning across the entire central dogma molecular biology. We literally demonstrated the first spatial multi-omics sequencing by combining a large panel of proteins with the whole transcriptome later on, and also the genome transcriptome, genome and the epigenome. In recent years, and there's a major breakthrough in spatial proteomics analysis, including what Professor Matthias Mann and Max Planck demonstrate this called the DBiT, and also our panelists, Dr. Zhao demonstrated in Cell just earlier this year can actually map out entire tissue spot by spot using mass spec, unbiased mass spec proteomics. So another layer of the central dogma molecular biology, not directly depicted over there, but the actual serial function. For example, metabolic function, we're working together with Professor Lin Yanxi at UC San Diego to bring in the cutting edge optical, metabolic, and chemical imaging tool in conjunction with our spatial omics to not only map out the cellular function directly in complex human tissue specimens, but you can link that to the omics across all the layers of the central dogma. Just a very brief history about the sequencing-based spatial omics. And Professor Joachim Lomberg, as depicted in the previous slide, is really a pioneer in the whole field. He came up with this concept. You can use spatial barcodes to tag the transcripts in the tissue you capture, but capture over the surface of a DNA microarray. And afterwards, you incorporate those barcodes and the spatial barcodes into the cDNA, the complementary DNA, such that when you sequence using parent high throughput NGS sequencing, you can read out where the transcripts are coming from, and you can also read out the spatial location. So we were kind of thinking about the same question around the same time, but we came up with total opposite approach. Instead of capturing, we send those spatial barcodes direct into the tissue. Turns out that's much more versatile, and the tissue matrix itself is the reaction chamber. You can design all kinds of chemistries to proteins, RNA, and also the epigenetic features, which tells you more exciting biology rather than just which genes, which proteins are expressed, but you know what controls the expression of those genes or proteins. All right, so over the past couple of years, we published a number of papers, including the ones really pioneered by my former postdoc, now also panelist here today, Professor Yan Deng from University of Pennsylvania. And if you're interested in the field, you can check out this review article we published a year and a half ago. And as I said, Nature Methods back to 2020 choose the spatial transcriptomics as the method a year. And in the same issue, our work was chosen by the editorial office to highlight the progress of multi-omics sequencing goes spatial now. This year, everyone got so excited as well because spatial proteomics was highlighted as the method of the year, and I was kind of honored to write a perspective article emphasizing the spatial protein profiling needs to be placed in the context of multi-omics to really understand not just which proteins are there, really need to understand the mechanism underlying the protein expression and also how proteins are controlling other layers of the central dogma of the molecular biology, which has been kind of underappreciated in the whole field. So I mentioned Professor Xiaowei Zhang and Professor Yukin Lomberg wrote the perspective article highlighting the two distinct approaches towards spatial omics, in particular spatial transcriptomics. Each has its own pros and cons, but we were thinking, can we really combine both? And you can get the benefit of the best of the both worlds, right? We started to explore combining the high-plex protein imaging, such as the technology called a codex to image tens or even up to over 100 proteins at a single cell level. What you are seeing here is the 53-plex imaging of the human lymph nodes, and by the same tissue, what we demonstrate is you can actually perform unbiased genome-wide spatial gene expression analysis using our DBiT spatial transcriptome sequencing. In the past, what people have done based on the spatial gene expressions, you can perform clustering, very nice clusters corresponding to different tissue regions. But by integrating the spatial transcriptome spot by spot with the reference data, you can infer the composition of different cell types in each spot and generating kind of pie chart associated with every single spot. But in our case, you don't need to just generate a pie chart because you do both on the same tissue, every single spot, you know exactly what type of cells you have. You can split the genome-wide gene expression profile into individual cells guided by the high-plex immunofluorescent imaging. So it's the first time you can really combine the best of the both worlds to construct the truly single-cell spatial transcriptome, or similarly can construct spatial single-cell spatial epigenome using the same approach, getting the best of the both worlds by combining imaging and the sequencing-based spatial omics. Now we are applying this approach to construct a map of human lymph node organ during the normal aging process to understand how the structural, cellular, and also the underlying biology, because we have the genome-wide transcriptome epigenome data to teach you the biology and the molecular biology, and to understand the aging of our own immune system. And also, if disease occur in our immune system, for example, in lymph nodes, starting from the kind of sort of the nodule and localized to more diffuse malignancies such as diffuse large B-cell lymphoma, can we map out the spatial temporal dynamics and a single cell level, cell by cell, across the entire lymph node, almost the entire organ in this case. As I mentioned, we're also excited about what actually happens at a functional level. For example, metabolism. Can you connect the cellular function, cellular metabolism, cell by cell to the omics? What we have demonstrated here is the high-plex immunofluorescent imaging to measure tens of proteins to kind of identify different cell types. As shown here, just 3 markers. But every cell type you'll see here, okay, the T cell, different B cells in the dark zone, light zone, and also like a follicular dendritic cells and macrophage in the tissue in the lymph nodes. Every cell type, but not every cell you see, you have the metabolic activity imaged by the cutting-edge optical technique called stimuli Raman scattering microscopy, established by Professor Lin at UCSD. What you see here is the redox activity, which corresponds to how the cell is going to utilize the glycolysis versus oxidative phosphorylation to power the energy production in the cells. But also you can look at many other metabolic pathways, such as lipid saturation, unsaturation, the protein amino acid metabolism, or the nucleic acid metabolism, and so on and so forth. Now, every single cell, you can also connect omics to metabolic activity and function, and also potentially can look into the dynamics of the metabolism as well. We are applying this to the study of the temporal process of the human lymphoma development, and we found a very interesting thing. For example, in normal lymph node organ, you see a well-balanced, you see a good balance between the 2 different metabolic pathways, this redox versus lipid metabolism to provide energy source to the cells. But in cancer, in lymphoma, both metabolic pathways are upregulated. That does make sense because I think the cancer cells require much more energy consumption, much more energy supply to drive their kind of malignant proliferation and growth and eventual progression to more aggressive disease. Okay, back to spatial transcriptomics. Turns out that's while my lab is very interested in spatial multi-omics to understand the biology, not just the cell type, but I would say the spatial transcriptomics to define the cell type remains the mainstay. And so I ask the chatbots again, so what spatial transcriptomics is, even though I have my own definition in my mind. But seems like the ChatGPT and the Gemini both can tell you reasonably good consensus understanding of what spatial transcriptome is. Two keywords. Number one, you look at the RNA, but you pretty much just look at gene expression. Indeed, that's where we are, even after 10 years of spatial transcriptomics development in the whole field. But turns out RNA is not just a messenger RNA, and also RNA is not a kind of static expression level you want to probe using the current spatial transcriptome technology. Every RNA molecule has very rich life cycle, starting from the transcription to the splicing process to give rise to mature messenger RNAs, and then some of them are going to give you proteins through the translation process and then degradation. This life cycle is controlled by many other non-coding RNAs. Actually, those non-coding RNAs seems like they are not important. Turns out actually they are more important because they are in the biology. They are regulating the function of the messenger RNAs. All right. Unfortunately, I can none of the spatial transcriptome technologies nowadays are really telling you this rich RNA biology, this rich RNA molecular biology. And we started to think, can we really do more than just a spatial gene expression? And also, can we do that in the human tissue, human organs? Because the study of human biology should be grounded in the tissue and organs of the human biology, as eluded by the father of the medicine, Hippocrates, back to 1,000 years ago. And so very interesting. I think the human tissues can be well preserved. I think Egyptians mummify human tissues thousands of years ago, even like 7,000 years ago. And some of the tissues still contain interesting biomolecules you can look into to understand some molecular biology. And this interesting news in Science a couple years ago, I think last year, uh, showing some discovery of the chemically preserved brain tissue thousands of years ago, and you can still feel it's like a jello-ish. And that's how nowadays we preserve the human brain. It's one of the largest, the human brain tissue bank in Boston. And what's the trick to preserve the brain, even at room temperature? That's done by its molecules of formalin. And it turns out in the clinical tissue specimens, that's also uh, the chemistry utilized to preserve the clinical tissue specimens. But afterwards, if you want to have the perfect tissue morphology, histological features will preserve, and you'll further embed then in the paraffin blocks, in the wax blocks. So this is called a paraffin formalin-fixed and paraffin-embedded tissue blocks, or FFPE. That's the most abundant tissue specimens in the clinical tissue bank around the world, and that it's also the best tissue specimen for our pathologists to tell you the precise, the accurate diagnosis for a variety of human diseases. But even our pathologists, I think they are really my superheroes. They just look at the tissue histology, they can tell you so much, but they still need way more molecular information stacked on top of every single tissue slide to do even better job. They want even more superpowers. But how to do that? As I said in the past, that has been very challenging to do anything from molecular biology level understanding cell by cell in this type of tissue in the formalin fixed. You can see the tissue morphology, but hard to do more understanding, more characterization of the molecular biology. I was asking my own lab members, and can we make that happen? Really change the landscape of molecular biology research for human kind of human clinical tissue research. And so, first of all, can we understand all those different RNA molecules to understand the rich RNA molecular biology first? All right. So it turns out some trick uh, uh, that was developed by Professor Steve Quick a couple of years ago to probe different RNA molecules in single cells using relatively low throughput single cell sequencing, but also now possible to do with paraffin-fixed FFP tissues. But the chemistry might be very useful. That's the polyadenylation of all different RNA molecules. I think the chemistry being used nowadays in single cell and the spatial transcriptome sequencing is largely based on the messenger RNA, polyadenylated messenger RNA capture and the downstream reverse transcription and the CDN synthesis. But if you add a poly-A tail to all kinds of RNA molecule, now you can utilize the same chemistry to probe the rich biology of all different RNA molecules. We say, okay, can we give it a try? But you need to make that chemistry work in situ in the tissue specimen itself, rather than you need to extract the RNA molecule. Turns out that's the step you're going to damage the RNA molecules, and you no longer have the high quality data from FFPE. Turns out that's true. And so now we try to do in situ polyadenylation, and afterwards we use our microfluidic device to send in the DNA barcodes, the spatial barcodes into the tissue, similar to what we demonstrated 5 years ago for spatial multi-omics sequencing called a DBiT. But in this case, we were able to barcode not just the messenger RNA gene expression, but we were able to barcode all kinds of RNA molecules, including the protein coding messenger RNAs and the non-coding RNA molecules. As demonstrated in this data, all kinds of RNA molecules can be spatially mapped, including I think in the microRNA and the ribosomal RNA, snow RNA, and tRNA, and also the non-coding RNAs. For the tRNA, you can actually see all kinds of tRNA molecules. tRNA, every single amino acid associated tRNA molecules, not just the total amount of tRNA molecules. And the joke I often say to my students, I used to drink a lot of Red Bull. If you pay attention to the nutrition fact in the Red Bull, they're putting a lot of taurine, which is good for your heart, for your muscle. But I always wonder if I drink a lot of Red Bull, in addition to taking a lot of caffeine, I take in a lot of taurine, but where the taurine goes. So now you can actually map down the taurine, tRNA, and that means, okay, eventually where the taurine gonna be kind of transferred by this tRNA to synthesize the protein. So where the taurine goes to power your protein metabolism. And also, tRNA was the first non-coding RNA discovered and recognized by Nobel Prize. And so I also mentioned that since we are able to tag the RNA, including on a kind of fragment RNA, turns out we can get even better data quality compared to conventional spatial transcriptome sequencing, in which you can probe just the 3' end of the messenger RNA. But in our case, because messenger RNA is highly fragmented, when you add a poly tail, you can reach the full length of the messenger RNAs. Number one, you get a much better data. You get a much higher quality data, as demonstrated in this kind of mouse brain tissue to benchmark against many other methods, including a probe-based spatial transcriptome, which I thought no way to do better than that. Also, you can sort of access the different splicing isoforms since you map down the entire full-length RNA rather than just a 3' end. So now we can look into different splicing isoforms of the messenger RNAs across the entire tissue in different tissue regions of the mouse brain. And for example, this splicing isoform protein phosphatase 3 CA subunit, if you look at gene expression, you do see expression across multiple brain regions in the hippocampal dentate gyrus, in the CA1, and also in the isocortex. But this particular skipping isoform is only present in the dentate gyrus. I don't know the biology of that, but at least if you don't have the technical capability to resolve different isoform, you will never discover, wow, the skipping isoform must be very unique for the biology of the dentigeral neuronal cells and not other cells in the brain. That's way more interesting biology now you can look into as compared to the conventional methods that only tell you which genes are expressed, which genes are not. The ultimate power to unlock human biology research needs to be done with the kind of archival human clinical FFPE tissue samples. Can we apply this technology to the human clinical tissue specimens as well? I want to use one specific case to demonstrate the power of this and the actual other techniques we are developing in the lab to make human molecular biology accessible to the clinical tissue specimens. This is a case of a patient, a 56-year-old who had a kind of stomach kind of upset for several months and came to Yale New Haven Hospital, and two biopsy samples collected in the stomach from this patient using endoscopy, and diagnosis was done by my close collaborator, my pathology colleague, Mina Su. I think one of the tissue biopsy look like this, and the other has a much larger lesion. And my pathology colleague, Dr. Mina Xu, given a diagnosis, all right, the first biopsy indicates that's a relatively lower grade lymphoma called a MALT, mucosal-associated lymphoid tissue. And the second does manifest some diffuse large B cell morphology, and they have done many other molecular analysis, and actually pretty expensive. Think about you do many other genomic molecular diagnostic testing. Eventually, you'll arrive at the conclusion this, wow, this clonal kind of evolution, clonal transformation of this low-grade lymphoma to high-grade lymphoma. This patient needs to be treated right away. All right. So if the patient develop this transformation, which is relatively rare, but if that happens, the prognosis is much worse, and you have to treat right away using chemo. Some of those patients might respond well to the targeted therapy, but you don't know who's going to respond to that therapy, who's not going to respond. Unfortunately, nowadays no one knows, and the bulk molecular characterization cannot tell you that at all, right? So we decided that patient is still alive, I think, fortunately. We hope we can do some deep spatial molecular biology analysis of those two tissue specimens to understand the biology of that particular patient. And fortunately, uh, if we can understand that, maybe after a couple of years, if the patient relapse, we can have much better understanding how to treat that patient using targeted therapy. Okay, the spatial transcriptome data gives you a very nice spatial clustering result, and all different clusters correspond to different tissue features, and those are the lymphoma region. And you can look at all different kind of cell type based on the gene marker scores. And but what is super cool nowadays, there's amazing opportunities that started to emerge in recent years, given the major breakthroughs in AI and machine learning is you can now take this high resolution tissue histology image. My pathology colleague can understand, teach you a lot of interesting things, but now can you incorporate that knowledge and the spatial transcriptomics to construct this almost truly single cell spatial molecular biology atlas, in this case, our molecular biology atlas, using the computational pipeline called iStar, developed by a friend of mine, Professor Mingyao Li at UPenn. And with that, you can, so now we also apply this approach to the higher grade lymphoma tissue specimens, as indicated here for that same patient. There's our original spatial transcriptome. There's AI machine learning, enhanced spatial single cell molecular biology atlas. Now you do see truly single cell resolution spatial atlas of the whole transcriptome, including all non-coding RNAs as well. So much information now you can give to my my pathology colleague Mina Shuti, potentially better understand that patient and better treat that patient. Just to show you one example, right? So from the small tissue, you look at where the innate immune cells are, the macrophage turns out enriched here. But in the diffuse B-cell large lymphoma, you see they're in total different location. They're very close to the tumor. Since it's a genome-wide data, and including many other non-coding RNA, you can ask a lot of biology questions. For example, using the Patho analysis, you'll see those macrophages are way more kind of M2 phenotype, and you can also see other metabolic pathways, either upregulated or downregulated in this sort of more aggressive lymphoma, and that might indicate how you can utilize those targeted or metabolic kind of targeting approach to better manage the disease or better treat the disease, either kind of targeted therapy or combination therapy. All right, so speaking about cancer, you have to think about the genome, the genomic alterations as well. In the past, that's not been possible, no matter single cell or spatial, to understand the sort of the single nucleotide mutations, variants directly in single cells or the entire tissue spot by spot, and also on the genome scale. Since our data has the full genome-wide coverage and full transcriptome coverage, for gene length coverage as well, we were asking, can we take advantage of that for coverage to map down the single nucleotide variants to some degree? That means the tumor mutational burden. That's a FDA-approved clinical biomarker to stratify patients for immunotherapies. And if we can do that, we basically can give you spatial resolve the tumor mutational burden. That's what we demonstrate. This accumulated as a VCON to some degrees, kind of telling you the tumor mutational burden, but now you see tumor mutational burden spot by spot by spot, cell by cell. And indeed, a high tumor mutational burden region corresponds to the tumor as indicated by the markers. But if you don't know the tumor markers, for example, for rare cancers, this should be your ground truth, because you see the genomic alterations and the genomic changes to drive the development of the cancer. Since there's a genome-wide mutational profile, now you can take that profile to further subclassifying the tumors into different clones using the genome-wide mutational profile, and to construct this tumor evolutionary dynamics, this tumor evolutionary phylogenetic tree, and those are more aggressive tumors, those are less. And so now you can visualize where they are. Okay, those more aggressive tumors and those tumors are slightly different phenotype. This first time you can directly visualize, all right, there's a tumor clonal, heterogeneous clonal dynamics in the human tissue, in particular human clinical FFPE tissue specimens. Now you have much better idea when the tumor actually develop, progress, and further evolve in this specific patient in this tumor. You can visualize individual mutations, and since the genome-wide, for example, one of those, we found a very interesting, I think, enriched in all those lymphoma, in particular this two. And the literature indicates, wow, this mutation indeed can drive the lymphoma to more aggressive stage by harboring a memory phenotype that makes the lymphoma kind of more resistant to drug, and also more difficult and more aggressive and more resistant to drug. So these two tumor clones are very similar in terms of the SNV profile, but we can also look into another genetic alteration, which is copy number gain or copy number loss. We found, okay, these two clusters actually showed quite distinct copy number changes in specific loci of the genome. So now I can kind of use both copy number alterations and the single nucleotide variants to decipher this tumor clonal evolutionary dynamics directly in the tissue, in the same patient, the same tumor, to understand the whole history of the tumor development and evolution. All right, so next few minutes, I want to highlight another important aspect of the technology to understand molecular biology of every single tumor, every single human tissue is the 95% of the RNA actually non-coding. They are not coding the proteins, but they are actually telling you biology. Now we are able to access that 95% of the most interesting RNA molecules, including all kinds of those, no matter large or small non-coding RNAs. Can I show you one example in microRNA? Everyone in the RNA biology field got so excited last year because microRNA is another now Nobel Prize winning discovery of the small RNA molecule. And we look into all those microRNAs, we detect 1,800 microRNAs in that tumor. And if you study human microRNA, you know about 2,000 human microRNA molecules in our body. We were able to detect almost the entire pool of the microRNA molecules. So with this rich data, we detect the microRNA spot by spot, cell by cell, but you also know the gene expression. Now you can ask the question, how each microRNA molecules can, for example, enrich or depleted in the tumor, regulate their downstream genes like a messenger RNAs, or upstream, which messenger RNAs might regulate that microRNA expression. We found two of the MIRA-21, MIRA-155 are strongly enriching tumor, and this is non-onco MIRA, so onco tumor promoting RNA. But what about MIRA-155? We look into the genes upstream and also the gene downstream. Now, this rich data directly tells you biology, which we could not anticipate. It's so fascinating. Okay, upstream, all the genes regulating MIRA-155 from our data, we found they are enriched for this pro-inflammatory, this inflammatory signaling called kappa B. So this sort of like a persistent inflammation drives the expression of MIRA-155. And the downstream, this microRNA triggers the activation of the tumor proliferation pathways, PI3K-AKT. Maybe I mentioned a little bit about this patient and how to treat. I mentioned, okay, PI3K inhibitor therapy has been used to treat lymphoma, but we don't know who's going to respond, who's going to not. So now in this patient, we say, wow, there's a MIRA-155 pathways involved, and the downstream signaling is PI3K-AKT. Maybe this patient might respond to the therapy. So this is how we can learn biology, and also the biology might help better stratify the patient for precision treatment and potentially even develop targeted therapy. For example, if we can target MIRA-155, that might work for this patient, right? So this work has been published kind of a couple of months ago in Cell, and also chosen on the cover. And I was wondering why they chose our paper on the cover, because the issue was focusing on molecular biology. Now is indeed, now we're unlocking the opportunity, the new opportunity to really understand molecular biology in human tissue specimens, in particular, the most abundant clinical archive of human tissue specimen. I just want to show you now we can also learn such rich molecular biology and a sort of the epigenetic level, which epigenetics means how the gene expression being controlled by the changes other than DNA sequence, so above the genome, but what other mechanism control which genes are expressed or not. In the interest of time, I don't want to discuss the details, but just to tell you it works. But jump to the opportunities in the future unlocked by a variety of the tools developed by my own laboratory, but also many others in the world nowadays. You can apply the spatial single cell and the spatial multi-omics to look into rich molecular biology, right? So this is sort of the word from my own colleague at the Yale School of Medicine. Although the mouse models are tremendously useful, and but often turns out the mouse models, they are just some models, but human get diseases, and the mouse models cannot fully recapitulate the complexity of human disease. And so human clinical trials are actually the best experiments. They are much better than animal models, but unfortunately, most of the human clinical trials give you those kind of biopsy samples in paraffin blocks. You don't learn that much, and most of the clinical trials are flying blind. And if that succeed, that's great. But if not, you actually don't learn that much from that. But I want to say it's changing. And just last week, FDA announced this sort of, I think that's a big surprise to the entire field. They decided to gradually phase out animal testing for some drug development, in particular antibody drug development and immunotherapy drug development. And and some big pharma such as Roche, and they are very forward-looking that even kind of launching a whole institute of human biology research to look into and the human clinical trials, number one, but also develop the model systems such as organoids to model human biology and human disease rather than fully rely on animal models. I believe combining the organoids to study the human tissue biology and disease and rich single cell molecular, single cell and spatial molecular biology profiling tools as demonstrated in our own laboratories and many others. And now we're at a crossroad and to enter a new era of human biology research and potentially going to transform the precision healthcare and also transform the drug discovery in the future. I love this image so much, but think about that's just in the beginning. You can stack so much more information, including the epigenome and also potentially incorporate proteome metabolism and also perturbation if you are using the organoids. All right, so I know I'm running out of time. I would like to thank all my lab members, current and former. Without their devotion to science, I would not be here to speak to you and my founding support. And also thank you so much for your attention. I would like to say happy birthday to ICANX again. And any questions, I'm happy to answer, and also looking forward to the panel discussion as well.